Is this actually working? Just think it's actually working. That'd be pretty cool if it is. So we'll see. Right, what am I doing? No sound detected. So hopefully you can hear me and it's coming through on here. And that will therefore mean that we get this side as well as that side. I'm hoping this works. Hi guys, if you are joining me, I am just setting up. I'm hoping that this is working simultaneously on both my screens and on the TikTok board as well. So if you can see the information, then that's great. And I would say that some questions that some of the students are really struggling with right now that may help is to do with genetics. But obviously, if you have any requests, please do ask away any of the questions that you would like me to answer. Hi, Jemima. So I'm hoping that you can see it quite nicely on the board. Hi. Right, so I'm trying something different tonight. I'm trying to do both YouTube and TikTok at the same time. So we'll see if it actually works the way I want it to work. Do you have any questions that you guys would like me to specifically cover? If you do, please do ask away. Ibris, hi, how are you? How are we feeling? Andreas, I'm glad you're joining as well. Have you guys got any topics you would like me to actually cover specifically? Please do ask away. Otherwise, I was thinking to do some genetics. Hi, Andreas. But yeah, if you have any topic areas that you'd like me to cover, please do. Respiration, sure. Do we want to look at a question on respiration, Jemima, or do you want to just go straight into some content of it? What are you looking for? Is it a specific part of the respiration topic area that you want? Some content, if that's okay. Yeah, of course. Of course, that's okay. So if we're going content then, I have a funny feeling that if they are going to ask questions, they did a lot on chemiosmosis last year, and they did quite a bit on the photosynthesis that links together with it. So what I would say is if they're going to do any questions on it this year, they're not going to ask questions, but they will link it to the use of it as potentially a respiratory substrate, which would be where I think they might go. So what I'll start with is looking at practically how to use a respirometer. So if we were looking at the use of a respirometer, yes, good job. Okay, so one of the things I would say to you is the fact that when they do respiration, they're looking for, do you understand what it means when we talk about respiration? Hi, Tidima. Do you understand the different parts to respiration? So it's not just that respiration is respiration, it's the fact that you've got different parts to respiration, and that it starts off with the fact that you have glycolysis being the first part. So if we were doing this and linking it to all the different parts, let me just move you guys out of the way as well. So first thing to do is glycolysis. Then after glycolysis, we have got the link reaction. Then after the link reaction, we've got the Krebs cycle. And then after the Krebs cycle, we've then got oxidative phosphorylation. Okay, so if we understand that, what we can see then is the fact that the glycolysis to linked reaction to Krebs cycle to oxidative phosphorylation, what we're looking at with the respirometer then is we know that there's air being filled into this gap. Because there's air in this container, it's a closed container, therefore any oxygen that's going to go into my respiring organism, whatever that respiring organism is, that is going to be utilized as that last electron acceptor at the oxidative phosphorylation. So we know the last electron acceptor as oxidative phosphorylation is where that oxygen is going to be used for those respiring organisms. Then what we need to look at is, well, okay, if that is where the last electron acceptor is, then where is the carbon dioxide that's actually going to be absorbed by the soda lime or potassium hydroxide solution? Where's that coming from? Well, we know that carbon dioxide is going to come from the link reaction because of decarboxylation. And we also know that the carbon dioxide is going to come from the Krebs cycle. So if they're going to link this, if they're going to give you a respirometer and expect you to apply it to respiration, which is where I think they could go if they're not just doing a planner practical, then it will be about that application. Where are these things coming out? Where's the carbon dioxide going in and being used as the last electron acceptor? Where's the carbon dioxide being produced? The links in the Krebs cycle. It's that kind of detail they want you to go into. Now, if we're using small respiring organisms, we don't want to harm them. So that's why we need to ensure that we put a gauze. Now, what they also will say is they'll say to put some cotton wool as well, just to prevent that gauze from touching and therefore preventing the respiring organisms from touching a harmful substance. And then obviously you're going to measure the distance moved, which is going to indicate that oxygen consumption. So it could be that they ask you to plan the investigation, or it could be that they give you a set of results and they expect you to know that if you've got your 1 centimeters cubed of the capillary tube moving, then that capillary tube moving, let's try that again. 1 centimeters cubed of a capillary tube. Therefore, you should be able to figure out what the volume of oxygen is based on the distance that's moved. So if you are looking at a manometer, which is another type of respirometer that can be used, it's a more advanced respirometer in the fact that you've got 2 different boiling tubes set up, and the calibrated scale is where you're going to use that movement of the liquid, because obviously oxygen's going in, carbon dioxide is being absorbed. You're then got that movement coming down into the first one, which has your respiring organism. Now, this could be, for example, a small respiring organism. It could be a pea. It could be anything which is a germinating bean. It just depends what they want to put in there. And I do feel like germination is going to come in. So it could be that they talk about different germinating seeds that are put into a respirometer and how that then looks at the respiration rate of those different seeds, whatever they may be. So then you have got obviously using glass beads as a control for comparison, and the fact that you can calibrate it and do repeats, you need repeats so that you can then do calculations. And obviously, if they're going to do it, they could look at brewer's yeast and using some form of carbohydrate solution. So one thing I would say is when you are looking at this type of thing, do make sure that you are confident in what would your independent variable be, what would your dependent variable be. So if I'm going to change the carbohydrate solution, I'm going to give it glucose, maltose, sucrose. I'm going to give it fructose. I'm going to give it starch solution. I'm then going to measure the rate of the respiration of the yeast. So I'm going to have to make sure that I keep the temperature the same, so a control variable, the volume the same, the mass of the organism, and so forth. So do make sure when you're doing this, and if they give you about the diameter of the capillary tube, which I said to you before, that you understand how to measure the rate of oxygen uptake based on the volume of the cylinder they expect you to know. They would give you the diameter, and then you would have to put that calculation in. So obviously pi times my diameter, which is 0.1 millimeters, divided by 2. Now let me just, they've not, when I copy that across, it should show that as being squared times 3. And then it's obviously over 2 hours, so you divide it by 2. And that's why you would get the number, and then you would change that into a standard form, which is 1.18 times 10 to the minus 3. Okay, so I haven't missed Andreas, your question. If you could go explain the answer to question 14, paper 1, 2021. Let's go to that one first, and then Abira, I have seen your question as well, but just give me a second. I'm going to work my way through the questions that have come through. Okay, so 2021, you said 2021. And you want question 14, 2021. Where are you? 2021. Oh, my brain's not working tonight. I've been doing my sessions, right? What am I doing? 2021, 2021, 2021, paper 1, 2021. And you said question 14. Where are you? Question 14. Multiple choice. Ah, there we go. There's a respirometer question. Perfect. Like you wanted it linked, right? So if we go to the respirometer question, if I add that in here. Okay, so here we go. Here's the question. So we've got a respirometer, and we are being told some information. And the information that we're being told is the fact that we have got 5 centimeters cubed. So, oh, no, I don't want to do that. We've got 5 centimeters cubed. So let me just add this in. Oh, don't freeze on me now. There we go. So 5 centimeters cubed of potassium hydroxide solution in the left-hand tube. Right, okay, so I'm just going to draw that in. So they've put in here 5 centimeters cubed of potassium hydroxide? Then the student suspended basket above the liquid and placed 10 grams. So we've got the basket above the liquid, but 10 grams of respiring seeds. Okay, so here we've got respiring seeds. And so we've got respiring seeds. We've got 10 grams. Making sure that, oh, hold on 1 second. That has not shown it 1 second. Do that way. There you go. Now you can see what I have got here. And I don't know if this is actually working on YouTube anymore because of me switching on this, but we'll see if that works. Anyway, okay, so what have we got? We've got the respiring seeds, 10 grams, left-hand corner, potassium hydroxide, 5 centimeters cubed. Which of the following statement about this investigation is not correct? Right, it's not correct. So the level of the liquid in the left-hand arm of the capillary tube would decrease, move downwards with time. So it's saying that the left-hand tube, that's going to move downwards with time. Well, it's not, because if I've got respiring seeds, the respiring seeds are going to take in the O2. The O2 is going to then be used as the last electron acceptor, which therefore means carbon dioxide is already being produced. The carbon dioxide is going to go into the potassium hydroxide solution. So I would expect that to go that way. So in that case, I would say that that one is not a true thing for respiration, but that because that's what it's asking me, I would assume that that's the right answer. But if that's the case, I need to eliminate the wrong answers. So I need to make sure that I can eliminate those, because if I can't eliminate them, then I don't know the topic area that I'm talking about. So if we then look, the potassium hydroxide absorbs the carbon dioxide. Yes, it does. We know that because it's another thing of using soda lime or potassium hydroxide. So that is a correct statement for use in this respirometer. The rate of movement of the liquid in the capillary tube is a measure of the rate of respiration. That is true. As long as we know the diameter of the capillary tube, as long as we know the volume of a cylinder, we can work that out because we're looking at the movement of the time. The tap can be opened to equalize the pressure in each tube. Yes, so the tap can be opened, whether it's open to let air in or whether it's open to let air out, is going to allow for that calibration. So therefore that would tell me that the only right answer would be A. I would expect it to go up, Andreas, because what I am seeing is that the oxygen is going to be going into my respiring seeds, which are here. So if oxygen's going in, the volume of air, because the carbon dioxide is being absorbed by the soda lime, is decreasing, and therefore the air will be sucked in to replace the particles that have been utilized in the test tube, basically. Volume decreases, therefore pressure decreases. If the volume increases, pressure would increase in force. Yeah, so it would be the opposite way around. Okay, so what did you ask for, Amna? You said, could you go through 1D and 2D on the 2022 paper 3? Right, hold on. Oh, I just wanted Excel. I didn't want to do that. Let's go away Excel. It's not what I want. So I want 2022. But on, you said paper 3. Paper 3, 2022. Where are you? 2022. 2022 paper. Oh. And question 1D and 2D. 1DII. Right, hold on. 1DII. 1C, 1D, I. So let me just go. So this one that you're looking at, state one method for inserting the AC6 gene. Is that what you were wanting? Yeah. Okay. So why does it say somatic cell gene therapy? Hold on. Right. So let me have a read of the question first till I get my head around what it's actually asking, because that's the key thing. It's always important that whenever you do this, that you do take the time to read the information they've given you beforehand to see what it's saying and then go from there. So what have we actually got in this question? What have they told me previously? Have they told me anything previously? ECG, disorder, pathogens, type of disease, and then they've said future treatment, right? So the gene therapy is a possible future treatment for heart disease, AC6 genes for one of adrenaline cyclase. Suggest how using gene therapy to increase levels in the heart cell may improve the heart function. State one method for inserting the AC gene into the heart cells during gene therapy. So it's talking about inserting the AC gene. So we're talking about how would we insert it, by using a vector, or what are we actually using? If we're not using a vector, what could we use? We could use a liposome. So why can't we say somatic cell therapy? Because somatic cell therapy is us talking about us inserting a gene into a body cell. So the somatic cell therapy is the whole name that encompasses it. It's asking you, how exactly are you going to insert it? How are you getting that gene that you want into those somatic cells? So it is all of it that you're talking about is somatic cell gene therapy. It's specifically asking you, how are you getting that gene in? So therefore, you're going to insert it either using the vector, using a viral vector, for example, where if you use a viral vector, it is going to carry the gene and insert the gene into the host cell like it would do if it was its own DNA or RNA. Or you could use a liposome which is made up of phospholipids, which are the same stuff that are able to fuse with the membrane. So therefore that would go in there. Do you need to know about ACTH and what triggers it for the hormone? No, they'll give you information on that. Question 22, paper 1, 21 DII. 2022, paper 1. Oh, sorry, hold on one second. Anna, you asked for 2D as well, didn't you? On this question paper before I move on to the next one. Right, let me just zoom out so I can see. 2D, yeah? Was it 2DI or 2DII? Let me just put the whole thing across. 2DI, okay. Okay, so in this one, what have we got? We have got some blood vessels. They've stained tissue that includes 2 different blood vessels, L and M, and a substance labeled N. Okay. State whether L is an artery or a vein and give 2 pieces of evidence, okay, from figure 2.2 that allow you to reach a decision. So the first thing I would say to you is anytime you see anything to do with blood vessels, you should be thinking about, okay, so I have my artery and I have my vein and I have my lumen size. So my lumen size inside my artery is small and therefore has a high pressure. And then here it is wide and therefore a lower pressure. So that's the first thing I would automatically go to. Oh, has it? Oh, there we go. No, I probably just didn't change the screen. Sorry, that's me. I put it on there, but didn't actually change it. So then you've got the smaller lumen, the wider lumen. So now you're wanting to answer the question as to, well, why or how can I tell the difference between these two? What is it that I'm looking at? So if I were to draw the lumen inside here, I can see the diameter is quite small. Whereas if I draw the diameter in here, and if I go all the way around, even though it's squished, I can see that that's a much wider diameter. So therefore, because it's a much wider diameter, that would tell me that what I'm looking at is an artery, and it's an artery because of that lumen size. So we have a small lumen, would be my first bit of the evidence. The other thing is the fact that you remember when you're looking at these arteries and veins, your next thing is about the layers. If we're looking at the layers going around, we know that there are 3 layers on both the artery and the vein, but it's about the thickness of those layers. And so therefore, there are thicker layers in the first one compared to the second one. So if you look at the layers here, and then the next layer, we can see that there are thicker layers, specifically thicker smooth muscle and elastic fibers in that first one compared to the second one. So that's why you get the sort of squishing of it. Does that help? Does that answer that question? Good. Right. Let me now go. What was it? It was paper 1, 2022. Paper 1, 2022. And what was the question? Your question was 21 DII. 21 DII. On the same paper. Yeah, I'm gonna give me a second. Let me do this question and then I'll jump back to that one for you. 20, 21. 21 D. Right, so Abira, if you're still here, I am now going to cover this question for you. You said about 2022, paper 1, 21 D. Oh no, hold on, DI you wanted. I've done the wrong one. Give me 1 second. And that's about ECS. So it's asking you about the embryonic stem cells, and it's asking you about what it means for it to be pluripotent. Do arteries have more collagen than veins do? So when you're looking at the arteries and the veins, you need to consider, well, what's the role that it's got to have? It's got to withstand pressure. It's got to be able to support the blood that's been circulated inside. So if it's got to support the blood that's been circulated inside, it therefore means you need to ensure that the layers can withstand that. So I will answer your question, Abira, in a second. I'll just go back to this one just to answer B's question. So when you're looking at the lumen size, the artery and the vein, when you're looking at the difference in the thickness, and we're talking about the collagen is actually thicker in the vein than it is in the artery. And so therefore, what you have to understand is that that layer is thicker. And you could see that in the question. We'll go back up a second. Where was it? Here. Oh, that one there. You can see there's a thicker layer of collagen here compared to in the artery. And so therefore, there is a thickness, but it's so minute in the difference. So the artery has to withstand a lot more pressure, but it's got a thicker, look at the layer of smooth muscle. Look at the layer of the elastin fiber. So because of this thickness of the smooth muscle, it doesn't need as much support in the shape of the actual blood vessel. Whereas when you look at the vein, it's got a smaller elastin layer, it's got a smaller smooth muscle layer, and therefore it needs more structural support. So that's the reason it's different between the two of them. Okay, back to the question that you asked about why is it pluripotent? So the reason it's pluripotent is you knowing that it cannot, so key thing to note, it cannot produce a whole organism. So that's the first thing. It can produce a whole organism. It can, however, divide and then differentiate. So it can produce more cells. So because of that, what does what mean? I don't know, what do you mean by? Oh, so what I mean by it can produce a whole organism. So if we have a fertilized egg cell, so if we have a zygote here we have a zygote, which is a fertilized egg cell. So if I've got a fertilized egg cell, which is a zygote, that will then go through mitotic division, and that going through mitotic division will produce a little baby that's smiling, its little chubby legs, and chubby arms. Is why I don't do things quickly. So it can produce a whole organism. Now, when it goes through division, it will go from 1 to 2, 2 to 4, 4 to 8, and so forth. So you get that. So we go 1 to 2 to 4 to 8 to 16. Up until this point here, if these cells separate, completely separate, what you can get is that one cell, when it goes through separation, can then produce two organisms. So there we have now our identical twins. So if the embryo splits before it gets the 16 cell stage, that therefore is going to mean that what you have got is the whole organism is being produced. So it up to the 8 to 16 cell stage, these cells are totipotent. They have the potency to become a whole organism and any type of cell. Whereas when we get past the 16 cell stage, then some of the cells have already started to differentiate. And because they've already started to differentiate, what's going to happen is they are going to start to produce what's going to become the placenta, for example. So what you have is, if we were looking at us and the development, is you end up with the tonoblast on the outside, and then the blastocyst, which is the mass of cells on the inside, which becomes the fetus. That is your embryonic stem cells, and that's the ones which are pluripotent. This ball of mass. Ball of mass? Mass of cells, this ball of cells. You can tell it's getting late. Okay, does that answer your question? Abira, if you're still here, I hope that answers your question. Okay, Amna, you asked for 6A and 6B, still on which question? 2022? 6A, 6B, where are you? 4, 5. Yes, you do need to know it in detail. It is module 2. It is 2.6, and it's cell division, where you're expected to know the potency of the cell. So you are expected to know about embryonic stem cells and their potency. Right, 6A and 6B. No, that's 5. 666. All right, okay. I quite like this question, but at the same time, it's not a very nice one. Oh, hold on. Are you still wanting me to do question 6, or you now want me to jump to 22 and paper? Oh, you're telling me it's paper 3. Yes, this is the one that you want me to do, right? With the CRISPR and the hemoglobin and sickle cell anemia. Yeah, perfect. Okay, so what do we have? Well, hemoglobin is an important protein, right? So they've told me already in the question it's a protein. So straight away, which we already knew anyway. Oh, okay. Which question is it? Okay, it's fine. It's fine. They've told you it's a protein. They've told you sickle cell disease is causing crumbs, wrong one. Abnormal hemoglobin. So we've got abnormal hemoglobin with this sickle cell anemia. A treatment is through using a CRISPR gene editing. I actually shared a video which was really cool about CRISPR genes editing the other day there. So what we have here is they've given us some information. You're going, oh, my teacher hasn't taught me anything to do with CRISPR or gene editing. I don't know this at all. You don't need to know it. The question and the stem of the question will give you the information that you need to know. They will be expecting you to apply your knowledge to be able to answer the question, because the question is saying traditional genetic engineering methods. Use your own knowledge, which you should know on traditional genetic engineering methods. Now compare it to what's in front of you with the information that they've told you. So whenever you get a question like this, it's important that you say, right, well, what have they asked me and what do I already know? So I'm just going to start writing about genetic engineering. What do I know? I've got some keynotes that I want to say. So I know I need restriction enzymes. Those restriction enzymes are going to cut. I'm going to need to use ligase to join my fragments back together again when I'm inserting the gene. I'm inserting the gene when it comes to genetic engineering, and that's then going to be transcribed and translated. So I'm using restriction enzymes to cut a gene. Oh, I'm using ligase to join DNA and a plasmid. And then that's my inserting of the gene. And that's going to be transcribed, translated. Right, okay. That's the try that again, traditional genetic engineering methods. So what have they told me here? Right, bone marrow stem cells are removed. We're going to remove the cells. An enzyme, so they're using an enzyme as well. I've got enzymes here, added to the stem cells. This enzyme is deleting bases, so that's deleting, whereas I'm inserting. The BCL11A gene usually switches off the fetal hemoglobin in adults. The gene editing stem cells are placed back. So they're placed back into the patient, and then the patient is now producing the protein, so therefore the transcribed and translated. So when it says describe the similarities and differences between the two, that's what it's looking for you to do. It's looking for you to do that comparison between those two different parts. They're both using enzymes to cut. They're both changing the base sequences. One's deleting, whereas one is inserting. Whenever we use a plasmid, normally we want to have a marker gene. So that would be what he's looking for with that one. And then what was your other question you had on this one? B with it as well. Let me just look what's B. Okay. Okay, so in this one is checking your understanding of what happens to hemoglobin when oxygen and also other things get involved. So the first thing I would say to you is think to yourself, right, they've talked about hydrogen carbonate ions, they've talked about hydrogen, they've talked about chloride. So I would straight away be going, right, H2O and CO2 goes to H2CO3. H2CO3 is going to dissociate into hydrogen and hydrogen carbonate ions. That hydrogen carbonate ion is going to be shifted out and chloride ions are going to be shifted in. The hydrogen is going to combine with hemoglobin, therefore releasing the oxygen. The oxygen can then diffuse out. So therefore, because the hydrogen ions are not involved in the chloride shift, the only thing that's involved in the chloride shift is your hydrogen carbonate ions and your chloride ions. That's the chloride shift. You're shifting one for the other. And the reason you're shifting one for the other is because you've got to keep that electrical chemical gradient correct. So wait, but they are right because they bind with hemoglobin. The hydrogen carbonate ions don't, the hydrogen ions do. So the hydrogen ions aren't involved in the chloride shift. The chloride shift is so that the hydrogen carbonate ion is moved out so that it won't recombine with the hydrogen ions. Because the reactions are reversible, and it's funny because someone just said, well, this is chemistry. Remember, these reactions are reversible. And because these reactions are reversible, it therefore means we need to remove the hydrogen carbonate ions out, because if we don't remove them out, then they will interact together again. So it was looking for, when you answered this question, it was actually looking for you to say the hydrogen binds to the hemoglobin. Excuse me, it is a product of the dissociation of carbonic acid, because this is my carbonic acid. And then what we were looking at is a hydrogen carbonate ion is a product of the dissociation. It is involved in the chloride shift, and it does have a negative charge, which is why I wrote the negative up there. No. Chloride shift is only the chloride and, excuse me, the chloride ion and the hydrogen carbonate ion being switched the other way. And then the chloride has a negative charge and is involved in the chloride shift. What's the whole thing called? The whole thing is, I guess it would be the reaction of carbon dioxide and water. So it could be the dissociation. So when we talk about the Bohr shift, and we talk about it shifting to the right because the partial pressure of carbon dioxide has increased, that's then resulting in the reaction going in the opposite way. Good. I'm glad that that has been helpful for you. I'm jumping around for those of you who are on my YouTube channel. I wasn't sure if it was actually, yeah, it seems to be lagging for some reason on YouTube because I'm also on TikTok tonight, so it's not really working as well as I'd hoped. For some reason. But what I'm going to do is I am going to finish now because it has been a long night. I will be back tomorrow at yeah, probably it would be at 8:30. Tomorrow should be a good time for me to come back at 8:30. I'll be on again tomorrow at 8:30 if you want to join me again and you have specific questions. Otherwise, have a good evening and I will see you